Internal fertilization. Fertilization occurs within female reproductive tract. Example terrestrial animals. Copulatory sex organs, penis or hemipenis. Benefits: high sperm count is maintained. Sexual theory is applicable. In some species, sperms are released in form of packets known as spermatophores. Example spider, scorpion. After fertilization, there are post fertilization events. On the basis of location of post fertilization, embryo development: oviparous, viviparous, ovoviviparous, ovoparous. Oviparous lay egg outside female body after fertilization. Example reptiles, birds. Egg laying mammals are monotremes like platypus, agdina. Egg large, having sufficient nutrient required for embryo development. Clutch size large. Protection of egg or embryo required. Viviparous. Fertilized egg is required within female reproductive tract. Embryo development occurs with uterus or womb. Live bearing or birth. Example mammals: Eurythenans, Methanans. Placenta for nutrient exchange, gas exchange, waste material exchange. Placenta, mother endometrium, and embryo chorion. Clutch size small. Proper incubation temperature. Embryo remain protected in female body. Ovoviviparous or pseudoviviparous. Fertilized egg is retained in female body but nourishment is obtained from egg itself. Example shark, anaconda, snake. Live birth. Embryo remain protected in female body. Proper incubation temperature. Placenta absent. Internal fertilization in mouse or human. Difficulties: fertilization occurs in oviduct. Difficult to study. Released sperms are at different stages of maturation. There can be multiple ways of acrosomal reactions. Events during fertilization: capacitation in ampulla of oviduct, movement of sperm toward egg, thermotaxis, chemotaxis, rheotaxis, entering sperm into cumulus, acrosomal reaction within cumulus, binding of sperm to zona pellucida or fertilization envelope, species specific interaction, sperm recognize ZP2 protein, fusion of sperm plasma membrane to egg plasma membrane, isomosperm, Juno egg, and CD9 protein, fusogenic protein. Block 2 polyspermy, fast block absent, slow block calcium mediated. Egg metabolic activation, resumption of meiosis 2, fusion of nucleus to restore 2N condition. Capacitation occurs within female reproductive tract, ampulla of oviduct, final maturation of sperms capable to fertilize egg. Major changes in sperm during capacitation: removal of cholesterol of albumin protein, lipid rafts, dissociate except at sperm tip. Species no specific protein on sperm surface are lost. These proteins block recognition site of sperm protein to zona pellucida. Potassium ion efflux. Potassium ions are transported outside from sperm head. Membrane potential become more negative. Increase calcium permeability. Calcium influx. HCO3 minus ions and other ions influx. Sperm adenylate cyclase Y. ATP to CAMP by SACY. Sperm adenylate cyclase Y convert ATP to CAMP and CAMP converted to pKa and that activate capacitation. HCO3 minus influx increase calcium ion concentration increase and these ions influx stimulate sperm adenylate cyclase Y. Calcium ion stimulate dynin and that cause hyperactive sperm. Increase of cytosolic pH 7.2 to 7.4, increase fibronectin expression. Then effect of increase of cytosolic calcium and pH of sperm. Increase of dynin activity, molecular motor ATP and associated with microtubule which form exoneme in sperm tail. Hyperactivation of sperms. Sperm rapidly migrate towards egg. Directed movement of sperm toward egg. Sperm flagellar motility. Dynein associated with ATP. Uterine muscle contraction. Thermotaxis. Heat sensing. A thermal gradient of 2 degrees Celsius between isthmus of oviduct and warmer ampullary region. Chemotaxis. Progesterone. Crisp 1. Sensing in rabbit. Sperm negative rheotaxis. Cues from the flow of liquid from the oviduct to uterus. Migrate against the direction of flow using cat spur. कैट स्पर है कैल्शियम ट्रांसपोर्टर स्पर्म कैल्शियम चैनल्स entering sperm into cumulus follicle cells which are loosely associated known as cumulus sperm move through intestinal space flagellar motion major acrosomal reaction occur within cumulus prior to binding to zona pellucida by Jin and colleagues fusogenic protein on acrosomal membrane. Acrosomal membrane fuses with sperm plasma membrane. Enzymes are released and acrosomal content is released. Digestion of component of zona pellucida and matrix of cumulus. Minor pathway 2. Acrosomal reaction occur after internalization of sperm with ZP after interaction of sperm with ZP3 protein of zona pellucida. Minor pathway minor or pathway 2 acrosomal reaction occur after interaction of sperm with zp3 protein of zona pellucida binding of sperm to zona pellucida layer species specific interaction sperm has zp2 receptor zona pellucida has zp2 protein gain of function experiment by Bakov et al. Transgenic mouse oocyte were constructed. They were expressing mouse zona protein and one of four human zona protein. Human sperm bind to mouse oocyte expressing ZP2 protein. माउस के प्रोटीन्स हैं ZP1, ZP2, ZP3. ह्यूमन के हैं ZP1, 2, 3, 4. Egg with ZP3 acrosomal reacted sperm and egg with ZP2 binding of acrosome reacted sperm. Fusion of sperm plasma membrane to egg plasma membrane. Equatorial region fusion. Sperm has isomo protein. Egg has mainly Juno protein and CD9 protein. Isomo immunoglobulin like protein interacts with Juno and CD9 protein. Sperm nucleus and centrioles enter the egg. Block to polyspermy. Fast block absent. Slow block calcium ion mediated. Start within a minute of sperm interaction. Slow, late, permanent block to polyspermy after first sperm interaction. Cytosolic calcium increase released from ER. Role of increased cytosolic calcium in ions. Activation of CAM kinase. V snare protein. Synaptotagmin activation. Synaptotagmin activation, vesicle fusion with plasma membrane, cortical granule exocytosis, release of ovastatin from cortical granules. Ovastatin removes ZP2 protein from zona pellucida because therefore no more sperms can interact. Loss of function in ovastatin mutation, then no removal of ZP2 protein and polyspermy can occur. The zinc spark. Release of zinc ions after entry of first sperm. Zinc binds to zona pellucida, zinc shield. Zinc inhibit acrosomal enzymes acrosin and MMP2 as well as enzymes necessary for capacitation. Acrosin MMP2 digest pellucida. सबसे बाहर होती है जोना पेलुसिडा इसके ऊपर प्रोटीन होता है ZP2 अंदर होती है फिर पेरीविटलाइन स्पेस फिर प्लाज्मा मेम्ब्रेन और अंदर की तरफ है जूनो प्रोटीन इज रिलीज्ड फ्रॉम oocyte सेल मेम्ब्रेन जूनो प्रोटीन क्या है सोल्यूबल जूनो बाइंड्स टू फ्री स्पर्म इन पेरीविटलाइन स्पेस एक्टिवेशन ऑफ एग फ्यूजन ऑफ membranes release of PLC zeta from sperm head PLC zeta enters egg cytosol PIP2 cleaved into DAG and IP3. IP3 allow release of calcium from ER to cytosol. Calcium activates calmodulin activated protein kinase CAM kinase. Removes inhibitor of mRNA translation. mRNA translation allowed. Resumption of meiosis 2. Egg is released which is arrested at metaphase 2. Cytostatic factor MOS inhibit APC. Calcium ions activate CAM kinase and that CAM kinase inhibit MOS. So now APC को कोई inhibit नहीं करेगा और meiosis 2 resumption. Egg metabolic activation. Multiple series of calcium waves generated. Increase in protein synthesis. Increase in respiration rate. Mitochondria number increase. Oxygen consumption increase. ATP increase and respiratory burst occur. Fusion of genetic material. Sperm nucleus highly condensed or compact. Protamines highly basic protein and high disulfide bond. Reduced glutathione present in egg cytosol. Break disulfide bonds. Uncoiling of sperm chromatin. Protamines are removed and replaced by normal histones and euchromatin forms. Fusion of genetic material. Each nucleus enter into G1, then S, then G2 and M phase. Nuclear envelope of both nucleus disappear and genetic material chromosomes collect at same place. Spindle forms and chromosomes arrange. Zygote has male and female nucleus entities are separate. Two-celled embryo, male and female chromosomes are in one common nucleus. Microinjection, male pronucleus because large size.